Hello and welcome to the Native Fuse Alliance Student Showcase. This is a seminar series sponsored by the NSF Includes Alliance, broadening career pathways in food, energy, and water systems within the Native American communities, otherwise known as Native Fuse Alliance. My name is Bernice Rodriguez, and I am the program coordinator for the Indigenous Resilience Center at the University of Arizona. The Native Fuse Alliance Student Showcase aims to showcase the outstanding work of our students to address food, energy, and water challenges in Indigenous communities and building curriculum to support Native American students. In addition, this showcase aims to connect students across the Alliance and provide mentorship, and support from the audience in attendance. This showcase occurs bi-weekly on Thursdays at 1 p.m. However, today we're wrapping up for the semester, so thank you for participating. So I'd like to introduce our speaker for today. Our student speaker today is Jesse Romero from the University of Denver of Colorado. Jesse will speak for roughly 20 minutes with a 15-minute dialogue. I will introduce Jesse Romero. Jesse Romero is a graduate student in the Department of Interactive Biology at the University of Colorado, Denver. She earned a Bachelor of Science and a Bachelor's of Arts from CU Denver in 2018, then completed the Environmental Stewardship of Indigenous Land certificate. Since she was an undergrad student, Jesse has been involved in multiple projects in the microbe ecological research lab of Dr. Timian Rowan. For today's talk, Jesse will describe her current research on how bacteria can degrade environmental contaminants. Today's talk is entitled Microbial, Community-Driven Degradation of 1,4-dioxin at the Lowry Landfill Superfund. Apologies, Jesse. I will now hand the platform over to Jesse Romero.
Thank you for the introduction. Again, my name is Jesse. I'm from CU Denver, and I'm going to be talking a little bit about my master's work here, and I'll break down what this title actually means. I know it's a mouthful for sure. And I think I'll go ahead and turn off my camera just so I have a good connection here. All right. So before I get into my research, just want to make sure that I took a moment to acknowledge all the people who are helping making this work happen. Of course, I have my advisors. There's several other students who are working on this project. And then there's also some funding entities and folks who have given us access to the site that I'm going to be talking about today. And then also just a special shout out to Dr. Timberly Roan. She's been a mentor of mine for several years now and has helped me grow, not only as a student and as a scientist, but also as a person in general. And so she's been super helpful, all of her guidance throughout the year. So I wanted to make sure that I give a special thank you to her in particular. All right, so for my talk today, this is an overview. So there's an environmental contaminant called 1,4-dioxane, and that's the chemical structure of it here. I'll be referring to it as dioxane throughout this presentation for short. And it's fairly prevalent throughout the environment, which is a big problem for us. But luckily, we're seeing that there's a lot of bacteria out in the environment that are able to take dioxane and get rid of it by degrading it to chemicals like carbon dioxide. And so they get rid of this contaminant just by breaking it down. And so they present a way to remediate sites that are contaminated with this particular chemical. And then here outside of Denver, Colorado, where I'm at, we have a site called the Lowry Landfill Superfund Site that shows lots of dioxane contamination. But that site in particular has been pretty successful at getting rid of that dioxane contamination using the bacteria that are naturally occurring in that site. So that's what my work entails, trying to figure out how these bacteria are so good at getting rid of this environmental contaminant. So I've mentioned dioxane a few times now, and so I'll describe what this chemical actually is. So dioxane It's a synthetic chemical. It's man-made. It's not naturally occurring in any given environment. It's also considered an inorganic chemical. And it's used a lot in different kinds of industrial processes. So in the past, it was used a lot to stabilize other kinds of chemicals. For instance, industries would have these big drums of chemicals, and to prevent them from degrading over time, they would sometimes mix in dioxane into those drums to preserve those chemicals for industrial uses. Those particular uses have decreased over time, but dioxane still gets used in other ways. And so oftentimes it's used as a solvent when we make products such as paints, antifreeze, shampoos, and dyes. And dioxane is also used sometimes as an ingredient in pesticides, food packaging, as well as food additives. And so dioxane is still used in lots of different kinds of products and industries. And so because it's so widely used, it gets released into the environment fairly frequently. And so what we're seeing is that a lot of industries, sometimes when they do these chemical reactions within these factories, a lot of the time dioxane will form as an unwanted waste byproduct, sort of by accident. And so those factories, sometimes what we'll see is that the wastewater coming off of those facilities will have some level of dioxane in them. And so that wastewater will then get into the groundwater and surface water, which will then eventually feed into our drinking water supplies. So that's one way that dioxane gets released from industries and then into the drinking water. We also see that factories that make certain kinds of products will sometimes form dioxane as that waste byproduct by accident. And then that dioxane will often end up in the products that we buy and we use in our everyday lives. And so some of those products are personal care products, as well as cleaning products that we use around the house. Just a few examples of products that will sometimes contain levels of dioxane in them are deodorants, cosmetics, shampoos, as well as soaps and laundry detergents. And the thing about that is that, if you take any one of these products and you look at the label for the ingredients list on the back of them, it's not going to say explicitly that it has dioxane in it, because again, dioxane accidentally gets produced a lot of the time when all of those products are being made in the factories. So we don't necessarily know for sure what products will have dioxane in them just by reading the labels, because that information won't be there. But there is a good chance that there is some amount just because of how those factories produce those different kinds of products. And so when we use these products at home, that represents another release of dioxane into the environment. A lot of these products will eventually go down a drain. And once that happens, that'll again release dioxane into a wastewater treatment plant, which can then feed into our drinking water sources. And not only that, dioxane gets released when we use these products and they go down the drain. Just the containers of those products as well can also have trace levels of dioxane associated with them. So anytime we toss out the containers of any of these products, that's potentially also releasing dioxane into the landfill that those containers are going to end up in. And so when we do that, dioxane is also, coming off of our landfills, getting into the groundwater and surface water, and then again, just circulating back into the drinking water that we use. And so dioxane is ending up in our drinking water a lot of the time. And this is a big issue because dioxane, in general, that chemical is very stable. It doesn't tend to break down when it goes through our wastewater treatment plant systems. And it's also very mobile. So once it gets into the ground, it has a tendency to sort of spread out and create these really large plumes that can sometimes measure a few miles, in length. And so again, dioxane, it gets released in all of these different ways. It's getting into our drinking water, and it's also spreading throughout the environment. And so there's a few risks associated with dioxane. I just mentioned all the different ways that it can get into our drinking water. Back in 2015, EPA did a survey of different drinking water supplies across the US and found that about 21% of them showed some measurable level of dioxane in them. And a lot of the time we see higher levels of dioxane in areas with high industrial activities. And some studies have also come out with rats and mice, exposing them to different levels of dioxane. And we're seeing that in those animals, sometimes they will develop kidney damage as well as liver tumors after getting exposed to dioxane. And, on top of this, right now in the United States, there isn't an enforceable federal maximum contaminant level for dioxane. So in other words, EPA doesn't have a limit that's set across the nation of, how much dioxane can legally be released into the environment. And so since we don't have that at the national level right now, states across the US have different limits of dioxane that can legally be released into the environment. But there's a lot of variation within those limits. Right now, they're ranging from about 0.35 to 77 micrograms per liter of dioxane. And there's also some differences as to whether dioxane can be released into groundwater, surface water. So there's just a lot of inconsistencies in terms of the limits that we're seeing right now across states. And something that's also developing is that some states like New York and California are also starting to implement some legislation to limit the amount of dioxane that can be present in personal care products. So New York just came out with some legislation, I think back in like December or something like that, trying to limit the amount in personal care products and cosmetics. And California is also working on similar types of efforts at the moment. And so obviously dioxane is a really big problem. But luckily, a lot of research is coming out showing how microorganisms can get rid of this contaminant using natural processes. And so what we're seeing is that a lot of these sites that show dioxane contamination, we can isolate different kinds of bacteria directly from those sites that show an ability to degrade that contaminant. And we're also seeing, some studies have come out that have found bacteria, from soils from sites that have never been contaminated with dioxane, and yet they still seem to have some kind of ability to degrade that contaminant. So microorganisms that are able to get rid of that chemical might be fairly widespread throughout the environment potentially. And so to date, there's been about 34 bacterial species and 3 fungi that show evidence of dioxane degradation. And they do this by using something called enzymes. And so if you look at this center figure here, what this is showing is a degradation pathway for dioxane. And so obviously it's really complicated, but really the parts that are important here to notice is that the dioxane chemical starts up here. Some enzyme will come in and start breaking it down into simpler and simpler chemicals until it gets converted into carbon dioxide. And so that's the process of biodegradation. And again, how this happens is that a bacterial enzyme will come in, it'll see that dioxane molecule, that enzyme will take up that molecule and then shred it up, converting it into some other kind of chemical. And so that's how biodegradation occurs. That's how these bacteria are able to naturally get rid of this and other types of contaminants. And so the site that I'm focusing on for my research is the Lowry Landfill that's here just outside of Denver, Colorado. So this is a landfill where back in the 60s and up to the 80s, a lot of industrial and municipal waste was stored at that particular landfill. And back in the day, at that time, all of the landfill pits that were used, they were completely unlined. And that was the standard at the time. That was legal at that particular point in time to have these unlined landfill pits. But over time, we saw that, of course, those unlined landfill pits weren't doing a good job of containing all of the chemicals contained within the waste that was stored at that particular site. And so chemicals started leaching out into the groundwater, and eventually EPA designated this site as a Superfund site in 1984. And then a little bit later, and so that prompted a lot of efforts to try to get rid of that particular contaminant, since it's obviously a potential human carcinogen. And so the way that this particular site is trying to get rid of dioxane is that it has a specialized wastewater treatment plant system on site. And so what this treatment plant system does, that's this picture here. What you're seeing is a really large bioreactor. You have to climb a ladder to get on top of this. And then if you look down, you'll see a whole bunch of contaminated groundwater here, sort of swirling about in these really big bioreactors. Within that water, you'll also see these little pieces of plastic here. We call them noodles because they look a little bit like noodles. What those pieces of plastic do, they basically just provide a surface for bacteria to grow on. And so what this is doing is that it's providing an environment for bacteria to grow. Once we can get that bacteria to grow, they can efficiently degrade dioxane and other chemicals that might be associated with the contaminated groundwater there. And so all of the bacteria in this system, they're coming directly from the groundwater itself. Nothing's added in. And this treatment plant in particular, we're seeing a dioxane removal efficiency of about 90% and above. And so it's really successful at getting rid of dioxane from the contaminated groundwater here, but we're not really sure why it's so successful. And so part of my work broadly is trying to figure out, who is degrading dioxane? In other words, what are the specific bacteria here that might be getting rid of that chemical directly? And also, how are they degrading dioxane? What kinds of enzymes are they using? And what does that tell us about the degradation process here at this particular site? And so to address some of these questions, if we look at this first one here, who is degrading dioxane? How we answer this is that we take those little pieces of plastic, the noodles that have bacteria growing on them, we extract DNA from those noodles, and then we sequence that DNA. Once we sequence that DNA, we can process that data and we'll get a figure that looks a little bit like this. And so this is a bar chart. Each bar represents a sample that was collected in the treatment plant here. Each of those colors that you see there are representing different groups of bacteria. And these samples, I think they were taken over about a period of 3 years or so. And so what you'll notice is that, we have some of these consistent colors here representing consistent specific types of bacteria that are constantly present in the system. And so as we collect more samples, we can see how, if at all, this changes over time, and whether or not we, see different kinds of bacteria present through the system. So that tells us a little bit about who is degrading dioxane, all of these different bacteria that you see here. Using similar sequencing data, we can also try to address the question about how they're degrading dioxane. And so again, bacteria are going to use enzymes to try to degrade dioxane. And so we can look for those enzymes by searching for the DNA sequences associated with those enzymes. And so we might get a figure like this. This is called an alignment. And so all of those strings of letters that you're seeing are sequences of DNA pertaining to different enzymes that might be participating in dioxane degradation. And usually when we get these kinds of figures, what we're looking for are regions that are similar across all of our enzymes. So just as an example, we have this one region here where there's sort of this red stripe and then this blue stripe. That's a pattern that we see a lot in these particular enzymes that degrade dioxane. So when we focus on those commonalities within these enzymes, those sort of give us clues as to the regions of the enzymes that might be necessary for dioxane degradation. And so when we take this information and we look at those commonalities across all of these sequences, we can then input that information into a figure like this. And so this is a phylogenetic tree. This is taking all of the enzyme sequences, and then it's basically sorting them in terms of how similar they are to one another. So looking at this, there's about like 5 groups here. And so they're all divided up based on how similar they are to one another. So for instance, if we just look at this one group here, all of the enzyme sequences there should be fairly similar to one another, but all the sequences in this group are going to be slightly different to this group, hence why they're sort of separated out like that. And so once we have this information about the similarity across these enzyme sequences, that can tell us a little bit more about how these enzymes are working to degrade dioxane, what are the commonalities across all of them that enhance dioxane degradation potentially. And so we can generate lots of hypotheses about how this process actually works when we get this kind of data. And then to wrap up here, just to reiterate the importance of this work. So hopefully I've convinced you that dioxane contamination is fairly widespread. This isn't something that's specific to industries and factories that are producing and releasing this chemical. You know, us, every time we use personal care products or cleaning supplies, we're also potentially contributing to this contamination problem, and we're also potentially exposing ourselves. And in addition to that, you know, anywhere you have a landfill, anywhere that you do have these factories or these different kinds of cleaning products and cosmetics and everything present, there's definitely a chance of dioxane contamination being present. And this is especially important when we're thinking about climate change and increasing water scarcity as well, because dioxane, we're seeing a lot in our drinking water. And so we need to make sure that we come up with a method that is readily available and that can efficiently get rid of that particular contaminant so that we can have clean drinking water supplies. So this really has implications for everybody because obviously we all need clean drinking water. We all use these products on a daily basis. So this really has some widespread implications. But luckily, we're seeing that microorganisms like bacteria offer a helpful solution to get rid of this particular contaminant. As difficult as this dioxane chemical is to degrade, bacteria seem to be, you know, the key to get rid of that particular contaminant from the environment. And so as we research this more, as we figure out what's going on at the Lowry landfill here in Colorado, that can give us some insight as to how to get rid of this contaminant from other similarly impacted sites. And so with that, I'd like to thank you for the presentation for this time today, and I'll be happy to answer any questions you might have.
Thank you, Jessie, for that insightful presentation. It is definitely interesting, and I'm sure your research, you know, everything you presented was amazing. So do we have any questions at all? We can open it up. If you'd like to just unmute yourself, you're more than welcome to and ask questions, or you can put them in the chat and I can go ahead and ask on your behalf.
I have a question, Jessie. So on behalf of all the people in the world who love water and appreciate water in all its forms, thank you so much for this work. It just gives me hope, and I really appreciate it. One question I have, this is not a technical question, but it's one that some of our students have. So for you working at that landfill and with the staff there and the leaders and the scientists or whoever, how is your relationship with them? Has that been easy or difficult for you to get in and be doing your research with them? If you could give us a little more information about how that worked for you.
For me, it's been fairly simple. I think it's really been, you know, my advisors, Dr. Rohn and Dr. Chris Miller here, who have really established that relationship over time. I haven't
Oh.
Jessie, I don't know if you can hear us, but you froze. There you are.
Oop, can you hear me?
Yes. Can you start your answer over again, please?
Okay, sorry
Thank
about
you.
that.
Thank you. That's okay.
So I haven't interacted with the site all that much. It's really been my advisors, Dr. Rohn and Dr. Miller, that have been establishing that relationship. For a few years now, they've been taking students over to that site to collect samples, to, get data like I was showing you earlier. And so it's kind of a reciprocal relationship because, we're at the university and we want to do research. And then, the Lowry Landfill, they want to know more about the bacteria at their site. And so, there's like an exchange of information going on there, but it's really Dr. Rohn and Dr. Miller that have been establishing that relationship and, just making sure that information gets shared. So it's really been their work and I've been just kind of along for the ride mostly.
Great, thank you.
And we have one question in the chat for you, Jesse. Kathy, thank you, Kathy Applin, for joining us. Kathy is asking, is there a difference for the bacteria regarding the location?
Let me see if I understand the question. I'm guessing this is about if there's different kinds of bacteria at different locations. Is that what you mean?
Yeah, kind of. I mean, is that bacteria kind of everywhere? And so can it work at other locations as well? Or is it that site?
Okay. So there are some specific bacteria that have come out in the literature that we see over and over again at, similar kinds of sites that seem to show dioxane degradation. So there are some that seem to be common across these different sites, but it is possible that at Lowry specifically, we could have a special, concoction of bacteria growing here. We don't know that for sure, but what's interesting is that the Lowry landfill, they've tried to take their bacteria that's, naturally growing at this site, and they've tried to give it to other places that are, trying to implement similar technologies to get rid of dioxane. And when they do that at those other sites, for whatever reason, the bacteria just don't really ever take off. They don't really degrade the dioxane successfully. So it's possible that there is something here at Lowry, whether that's another bacterial organism or maybe some kind of a nutrient that's in the area here that might be, giving rise to a very specific microbial community. So there is that possibility, but there are definitely a few specific bacteria that we are seeing consistently across these sites as well.
Thank you, Jesse, for that response. Do we have any other questions at all for Jesse? Again, you're more than welcome to unmute and ask your question, or you can add it in on the chat and we can, I can ask on your behalf.
If I may, Jesse, very interesting work. I wonder, biodegradation seems to be a very hot topic and promising and exciting, like bacteria that degrade some plastics. But based on your experience and the work that you have so far, what do you see as the major shortcomings or challenges of this approach to pollution?
Ooh, that's a good question. Well, I mean, I guess in general, not every site that shows dioxane degradation has the means to, construct a treatment plant facility like the Lowry landfill has been able to. Not everyone has resources to that or, the infrastructure in place. And so, that challenges us to think of other ways that we can get biodegradation to happen across different sites that might not have the technology in place right now to do that, at least the way that the Lowry landfill is doing that. So I think probably one aspect, one challenge would just be, the infrastructure and making sure that this technology is available where it needs to be available.
Great, thank you.
Is there any other questions we have for Jesse? I do have one question for you, Jesse. So one of your slides, you talked about the contamination and, just being more aware so that way, we're able to help the environment, right? So what can individuals do on their part? Is it something of just, when we're purchasing deodorant or just, the regular household items, is it something that we need to read on the label just to be more aware of what we're purchasing?
Yeah, that's a tough one because, for some of these products, it might not necessarily say on the label whether there's dioxin in there, because dioxin will sometimes just accidentally form while those products get manufactured. So yeah, it's a little bit tough to say. I don't, at the moment, I'm not sure exactly how, changing, our purchases is gonna impact, the release of this particular chemical. So I'm not totally sure at the moment, but I guess, another way to go about it is just to keep an eye on the legislation that's coming out. I'm sure that a lot of people, this is the first time that you're hearing about dioxin today in this chemical, but, I'm seeing it a little bit more in the news now. And of course, New York and California in particular are trying to get legislation to limit that chemical in certain products. So I would say, just keep an eye on the legislation and maybe see if it comes up in your state as well, because that might be a way to limit your releases.
Thank you for your response. So Dr. Chief put in the chat, she wants to thank you for your presentation today. And she also wants to know, what are your career goals?
That's a great question. I go back and forth with it. I'm thinking about doing a PhD, sort of continuing on with this work, because even once I wrap up this master's, I think we're gonna end up with more questions that we're going to ask than we have, answered. And so maybe I'll be able to address those in a PhD. So yeah, I'm still deciding. I might continue on with this research though. That's definitely a possibility.
Thank you again, Jesse. Do we have any other questions? Jesse, we thank you for your time. And again, thank you everybody for joining our series this semester for our student showcases. You can find additional information on our website at nativefuse.org. I put it in the chat for any upcoming programs and summer programs, fall programs. We're getting thank yous, Jesse, for your presentation today. Dr. Chief did have one more question: to pursue a PhD. Is that correct?
Yeah, I'm thinking about it.
Perfect. Thank you. Any last minute questions, comments at all, Jesse, either from you or from the audience here today with us?
Well, thank you very much for the time. I really appreciate it and hope it was insightful. So yeah, thank you very much.
Thank you. Thanks again, everybody. Enjoy the rest of your day.
Thank you. Thank you, Jesse.